Hey guys, so Dr. Stover here. I'd like to start off by apologizing for getting these videos up later than I'd intended. Um, Started having the headache last night, woke up this morning, headache was too bad to actually do anything. Um, and I've been fighting it, but for the most part, it's at least chill enough that I can do something. Please know that I am trying to take care of myself. I've been going to see a chiropractor, and that's kind of why I have a headache because the road to recovery tends to have some bad days, right? But I am way better than I was before I started seeing the chiropractor, so please know that I am on the mend, but still recovering, okay? So, but we're gonna pick up with disaccharides. All right, so we have 2 lectures left. So basically, what's going to happen is we're going to lecture today, Friday, and we will finish the lecture Monday, have a quiz Monday, and then Wednesday will be your exam. Okay, so be ready for that exam on Wednesday. The Wednesday exam will be on chapter 6 and 7. Right? But we'll have the quiz Monday so that you can get familiar with the system and everything that you're gonna have your test on Wednesday. All right. But as for today's lecture, we've already talked about monosaccharides, so now we need to get into disaccharides. And disaccharides are pretty straightforward. It's a linkage of 2 monosaccharides, right? By a glycosidic bond, okay? So where's the glycosidic bond? Well, I mean, it's this thing holding them together, okay? So this is your glycosidic bond. Notice the 2 different types of nomenclature, okay? So notice that you have this weird sort of Z-looking shape associated with this glycosidic bond. And then you have what looks like a U associated with glycosidic bond. Take a look at the nomenclature differences, okay? So when you see the Z every single time, this is associated with beta, right? And when we see the U, that's associated with alpha, okay? So that just tells you the type. You can recognize right away if this is alpha or beta based off that glycosidic bond, okay? Now I'm trying to get my mouse back. All right, so as with proteins, we're going to refer to each one of these as a residue, okay? So what lactose is one galactose sugar linked up with one glucose sugar. And notice the 1,4. The 1,4 is meaning, so like carbon 1 is linked up with carbon 4 in the other structure, and you're always going to get a planar type look whenever this happens. Notice how this one isn't planar. This would allow you to actually like stack up and almost create like a helices or something. This is a 1,6 linkage, okay? But whenever you see all of these are 1,4 linkages because they're flat, right? And then so sucrose, what sucrose is a glucose linked to a fructose. Maltose is a glucose linked to another glucose, so completely symmetrical. And then here, cellobiose is also glucose linked to glucose, but look, the only difference between these two is this one has alpha linkage and this one has a beta linkage, okay? And then this is also a glucose linked to glucose in an alpha linkage, but isomaltose, meaning it is an isomer of maltose because it has a different linkage. It's not 1,4. This one is 1,6. Okay? But basically what happens is this glycosidic bond is a hydroxyl that has interacted with another hydroxyl intramolecularly, and that's what's holding these 2 sugars together. Now, okay, except for sucrose, right? Sucrose is an exception, and we'll talk about this, but the other ones all have what we call a free anomeric carbon. We refer to this free anomeric carbon as the reducing end. Okay, so let's talk about anomeric carbons a little bit more. So what I'm going to do is I'm going to shoot over to a link I found because I know that I didn't require you guys to take organic chemistry. So this is what an anomeric carbon is, okay? So this is at Chem UCLA if you want to find it on your own. But the anomeric carbon is this. So in your sugar, you have a carbon on the end that is directly attached to, so you have a double bonded oxygen and you have a hydrogen attached to that carbon. This carbon has 2 hydrogens attached to it, is a single bond to an oxygen, and then has that hydrogen attached to the oxygen. This is what we refer as the anomeric carbon, okay? So this is this new stereocenter, right? This is the stereocenter, and we refer to the stereocenter as the anomeric carbon, okay? Because when we cyclize, remember 5 links up here, so this is our anomeric carbon. So if we were to look at some sort of, these are what shape? So it kind of looks like chairs. So this is our chair conformation. This isn't the boat, right? Because remember the boat would look like this. It looked like a boat, but this is our chair conformation. Okay, so this is your anomeric carbon. It's always essentially going to be, you're going to have an oxygen here and on one side of the oxygen is going to be the CH2OH, and on the other side of the oxygen should be the anomeric carbon. So if you go left and you see the CH2OH, then you know that it's the other direction, right? And it should always be here in this bottom right-hand corner. This is your anomeric carbon, okay? Now, and that's what this is saying. This is saying focus on, go to the right, right? We want to find the anomeric carbon. And if we scroll down, how do you know if it's alpha versus beta? Well, if it's pointed down, right? If it's pointed down, then it's alpha. If it's pointed up, then this anomeric carbon would be associated as beta. Okay, so beta versus alpha. And then this also goes into what happens. This goes into a lot more stuff than I really expect you to go into, okay? But there's some practice questions here as well about try to identify the anomeric carbon. So again, the anomeric carbon would be this one. And since this is pointed up, not down, it would be beta, right? Again, anomeric carbon is going to be here. It's pointing up, so it's beta. Anomeric carbon pointing up, again, it's beta, right? So all those are kind of similar, but here we have our anomeric. Here's our oxygen, which means this is our anomeric carbon, and the hydroxide is pointed down, so this would be alpha. Same thing here, oxygen. This is pointed down, so this is alpha. This is also pointed down, so this is alpha as well. The anomeric that's linking the two together. So now let's go back to the PowerPoint. All right. So what's going on here then is you see that basically what the free anomeric carbon, why do we call it the free anomeric carbon? Because the other one was used to link up. So yeah, this was originally associated with being an anomeric carbon, but it's not free anymore. It can't really do any chemistry anymore. The free end is always going to be written to the right. So what we mean is this is a reducing end. So the chemistry is going to happen here. The other end is not where the chemistry is going to happen. The chemistry is going to happen at this free reducing end. And then all of these structures, the other end is non-reducing end. So this isn't really where the chemistry is going to be happening. So what's interesting about sucrose is sucrose is not a reducing sugar, right? It's not a reducing sugar because it doesn't have a free anomeric carbon here. All right, so sucrose is very different than all of your other sugars. Okay, so what do I want you to be able to do? I want you to be able to recognize what an anomeric carbon is. I want you to be able to identify the difference between reducing and non-reducing. Know that sucrose is not a reducing sugar, right? We talked about that, how it is not a reducing sugar. Oops, sorry. We also talked about the difference between 1,4 linkage versus 1,6, how they look different. 1,4 is linear, 1,6 would be more of a stacked conformation. And then how to recognize the linkage in the center of the structure, right? If you see the little Z conformation, that's corresponding to beta. And if you see this U conformation, that is corresponding to alpha. Now think about it. Remember, because we said before, what did we say previously when we were talking about that PDF? Let's go back to it and look again, right? So if we go back to this PDF and we scroll up, when it's pointed down, it's alpha, and when it's pointed up, it's beta. Now compare that to these two structures, right? Down is alpha. This weird Z is supposed to be representing going up, so it's beta. So we're not reinventing the wheel here. I mean, it's saying the same exact thing. It's just a different look because it's connecting two sugars. All right? Okay. So now, so those are your disaccharides. So that's the linkage of two monosaccharides. What about polysaccharides? So polysaccharides, these are, long chains. right? They're also called glycans because they can be made up of monosaccharides or a derivative of another saccharide. And most of the carbs that we find in nature are polysaccharides, okay? So they can be a homopolysaccharide, meaning they only contain one kind of monosaccharide unit. So you could imagine here, this would be like a protein that was fully made up of glycine. So it's just like just not very interesting, right? Or you could have a heteropolysaccharide where it's made up of more than one monosaccharide, right? So it's got some glucose, it's got some fructose, it's got all different types of units, just like your peptides are made up of multiple different types of amino acids, right? And these polysaccharides, they can differ in terms of chain length, like how big they are, and they have branching abilities as well. So let's look at an example of a polysaccharide. So here's starch, right? Starch is very important. It's a plant storage protein. I mean, sorry, not protein, a plant storage polysaccharide. Oh, use an example of protein, and now I'm getting stuck on protein. Plant storage polysaccharide, okay? Most of your starch is this structure called amylose. And what amylose is this is glucose, and it's just repeating glucose. And this linkage looks like a U. This is alpha, right? This is an alpha linkage, okay? So it's an alpha linkage, which means basically you're only going to have one reducing end, and then you'll have your one non-reducing end. It's just one really long chain. So that's the smallest percentage of what starch is. What starch mostly is this. It's amylopectin. And amylopectin is basically amylose, except for it periodically every 12 to 30 residues will branch and then start a new chain and then branch and start a new chain. Um, so it can, and basically when it branches, that means that it can have multiple reducing ends or multiple non-reducing ends, whereas since amylose is just one long strip, it's a little bit of a different structure there. So let's look at the structure of amylose. So it's alpha amylose. We talked about how it's alpha because we saw that U-type linkage. It's 1,4 because it was a straight line, right? And this sort of structure, the chains can be varying in length, but these things are huge, right? And amylose itself is actually very poorly soluble in water, okay? Because if you look at it, there's not, I mean, you've got hydrogen bonding ability right here, but nothing else about this is really going to want to interact with water. So it doesn't really interact very well with water. So how it counteracts this is it actually sort of forms a helix, right? It forms a helix and it wraps upon itself. And what's interesting is I don't know if you've done this before, but if you've ever tested for the presence of starch, how we press for the presence of starch is we use a potassium iodide solution. Now, we don't care about the potassium. What we care about is the iodine because the iodine will actually fit down inside of this amylose structure. And when it does that, it gives it a blue color. So that's how we actually test the presence of starch is we add some potassium iodide to our solution, and if it starts turning blue when we add it, then we know that it's got starch in it, right? So just thought I'd tell you that in case you've ever done an experiment like that. But why is it mostly amylopectin? Why do we care more about the branching ability? Well, that's because the more branching you have, the more chance you have to come in here with phosphorylase and actually pop off these sugars. And because the whole point is you want to be able to pop off these little glucose units because maybe you need to quickly release them or store them, or you just need them right away. Like if your body desperately needs energy, it needs to be able to pop one of these suckers off and move it forward to metabolism. If it's all in one long chain, it can only release one at a time, right? But if it's multiple linkages all connected together because it's got all this crazy branching, you can start releasing multiple units at a time. So hopefully you see the benefit in having branching and starch because it really does speed up our ability to get a lot of energy. Otherwise, I mean, it would take you a long time to build up enough energy to actually be able to do anything that you needed to do. Okay? All right. So we will pick up in the next video with glycogen. Okay? All right. See y'all in a little bit.